Hi, everybody. Welcome to our final webinar in our 2025 webinar series, which we've called Mastering Lewy Body Dementia Together. My name is Keith Fargo, and I'm the Director of Scientific Initiatives here at the Lewy Body Dementia Association. This year, with you, we explored a range of topics to help you better understand Lewy body dementia and its symptoms, to help you strengthen your ability to advocate for services and support, and to pick up new tools and techniques to live better with Lewy. Next slide, please. Thank you. LBDA's mission is to optimize the quality of life for those people who are affected by Lewy body dementia. And to do that, we accelerate awareness, advance research for early diagnosis and improved care, and provide comprehensive education and compassionate support. Next slide, please. A few technical items I'd like to review before we bring in our presenter today. So first up, if you'd like to have closed captioning, you can launch that closed captioning by selecting the button or the icon at the bottom of your Zoom toolbar that says Show Captions. And you can use that Show Captions tool anytime during the presentation to turn captions on or off. I'd also like to draw your attention to another icon on the Zoom toolbar, which is called Resources. And in that Resources icon, you will find web links and downloadable resources to accompany today's session, which you can use today or if you watch the recording later. You are encouraged, please submit your questions throughout the presentation. We will have a Q&A period at the end. Chat is disabled in this particular webinar format, so if you have questions, please do submit those through using the Q&A icon. And I'll repeat, that is the Q&A icon because chat is disabled. And type those in, like I said, throughout the presentation, and we will address as many of those questions as we can at the end from that audience Q&A pool. My colleague Rose will be dropping links into the chat now, and also my colleague Glorious may be dropping some during Dr. Irwin's presentation. These links are also going to be in your email inbox in about 1 week from today as part of our post-webinar thank you email. This presentation is being recorded, and that email we send you in about a week will also have the link to view the recording along with those resources. And that'll be sent out to everyone who registered. So even if you can't stay for the entire time today, you will get that link to watch the recording later. When you leave the session today, a brief survey will appear, and it's going to ask you for your feedback about today's presentation. We really would greatly appreciate it if you would take a few moments to complete that survey. It truly does help inform us for our content for next year's series and beyond. I can tell you that I'm on the team that reviews those, and we do take them very seriously. So please do take the time to fill those out so we can continue to improve. Now, before introducing today's speaker, I am going to take a little bit of time to make you aware of some additional educational resources from LBDA. So next slide, please. You can keep learning about Lewy body dementia until our next webinar with our on-demand learning options. Recordings of all of our past webinars can be found on the YouTube channel, LBDA TV. Don't worry right now about trying to get this QR code on your phone, as my colleague Rose is going to put a link in the chat. Next slide, please. Also, the Lewy Learning Center offers courses on a variety of topics that you can watch, rewatch, and share with friends and family. And these are self-paced, self-guided learning opportunities. You can find the Lewy Learning Center on the Thinkific website. And my colleague Rose is going to go ahead and drop a link to that as well. Thank you. And next slide. You can also watch the film Spark about Robin Williams' battle with Lewy body dementia and many of our other video offerings on our Mediflix channel. That is at mediflix.com, and Rose just dropped a link to that in the chat as well. So let's move on to slide 7. And I would encourage you to think about joining our social media channels, because that's really a great way for you to stay up to date and in the know about upcoming LBDA webinars and programs. Don't worry again about trying to capture all of these links and usernames right now, because again, we will be sharing these slides with you in about 1 week, and you'll have the opportunity to take a closer look then. Next slide, please. And finally, a great source of information always is the LBDA homepage, where you can always find us. That is at lbda.org. Again, lbda.org. So now we're going to learn a little bit more about who's with us in the audience today and what your experience is with Lewy body dementia. So Glorious is going to launch a polling question that you can answer on your screen. Thank you very much. So this question, we'll give you a little bit of time to answer this, is how long have you been either living with LBD, if you're a care partner or a person with Lewy, or treating or learning about or advocating for Lewy, if you're a healthcare provider or a researcher, nonprofit organization member, volunteer, or advocate. And the options are 0 to 3 years, 4 to 8 years, 9 to 15, and in many cases, or some cases, more than 15 years. All right, we got some great responses coming in here. I'm going to go ahead and end the poll. It looks like we've got a pretty good selection and it's starting to stabilize. So sharing the results here, I hope you all can see this. Looks like we have about half, a little over half of the audience today has said they're fairly new, 0 to 3 years of experience with this disease. Another third said they've been at it for a while, 4 to 8 years. 12% said 9 to 15 years. So we do have some folks here who are fairly experienced. And then 4% who said more than 15 years. So I do want to recognize that there are those of you who are very experienced here today. You know, let's say 16% who've told us that you've been at this for 9 years or longer. But please do bear with us, because of course we do have that more than 50% of the audience who told us that they're less than 3 years in. You know, so we will be discussing things at a level that will be fairly understandable, hopefully, to all of those folks, even the folks who are new. So appreciate you sticking with us through that. I'm going to stop sharing that poll now. And then one more question that should help us, help our presenter really know a little bit more about today's audience. And this is, how knowledgeable are you on today's topic, which is research advances in the past year? So we'll give people a few moments to answer this. All right, thanks everybody for putting those answers in. It looks like we are starting to stabilize around a percentage here. So I'm going to go ahead and end the poll and share the results. Thank you. And all right, so 32%, so just under a third of you said that the topic is new to you. Just over half, 55%, said that you know a little bit about this topic. That's great to hear. And 30 joining us today, or just people who are really hungry and interested in research information and spend a lot of time taking a look at the research literature and what's coming out. And then 1% even who said they're an expert in today's webinar topic. So that is excellent to have you on as well. I'm going to go ahead and stop sharing those results now. Okay, so thank you again for sharing those results with us. We really appreciate it. And now I'm going to introduce our presenter, Dr. David Irwin. Dr. Irwin is an associate professor in the cognitive division of the Department of Neurology at the University of Pennsylvania. He is a cognitive neurologist who evaluates and treats patients with a range of neurodegenerative conditions, but with a specific interest in DLB. He has an active translational research program called the Penn Digital Neuropathology Lab, where he and his research team study human brains to better understand how brain changes lead to the symptoms of Lewy body disorders, including Parkinson's disease and dementia with Lewy bodies. Dr. Irwin is also a project leader for the NIH-funded program at Penn called the Center on Mechanisms Underlying Cognitive Heterogeneity in Synucleinopathy. That's a mouthful. In that program, he and his team investigate clinical and pathological facets of DLB. Dr. Irwin also leads the Penn site for the DLB Consortium and is involved in several areas of patient outreach and education. Please use your reactions tab to give a warm welcome to Dr. Irwin. Dr. Irwin, take it away.
Oh, thank you, Keith. Thank you for the introduction and opportunity to present and interact with everyone today. It's a pleasure to be with you. Today's talk will be about a review of research in the past year in 2025, innovations and insights. These are my disclosures. And I wanted to start briefly with an overview of the acceleration in LBD research. As you could see illustrated here, PubMed is a publicly available open source tool to query the medical literature of scientific work. And what's depicted here, it gives you frequency of publications or peer-reviewed scientific publications by year. And a query for dementia with Lewy bodies shows us a nice exponential curve of a growth of scientific contributions on this topic. And that doesn't include related topics to DLB, including alpha-synuclein and some of the mechanisms under the microscope. So there really is a lot of momentum in discovery for LBD, and I'm excited to speak with you about that today. And we have a big task ahead of us if we confine our search for the past year or so to review as we enter into the new year and reflect on what we've learned and where we're headed. We have close to 2,000 scientific reviews. So what I'll present is meant to be an overview and what we call a data blitz style presentation of high points in a theme related to advances in Lewy body disease that I hope is helpful. I look forward to the discussion at the end. And our overall outline will start with just an overview of terms and some of the biological definitions that are being used to describe LBD, as well as a highlight of the 2025 NIH Summit on NIH priorities for LBD, which are really important. And then we'll go through three categories or areas of research: clinical research, including clinical trials; translational research. This is research done with observational research, collecting data and samples to help inform clinical trials; and then mechanistic research, things looking at why Lewy body happens and how we can develop treatment targets. So one of the themes I'd like to talk about today is that LBD is a multi-system disease. What's depicted here is a photomicrograph or a microscopic image of a Lewy body in a brain cell. And what you can see, all the different arrows are the clinical manifestations that we see associated with this alpha-synuclein protein, a protein that we all have in our brains, that can aggregate into these Lewy bodies, these round inclusions and Lewy neurites, these other neuronal inclusions, and cause clinical symptoms. And not every patient gets all of these symptoms. And the management of Lewy body can be complicated and require a team effort of different healthcare professionals. And although this is a few years old now, is an important paper of Lewy body experts that got together and collated the evidence, because historically clinical trials in Lewy body disease have been less common than Alzheimer's, for example. But that's changing, and we're seeing a lot more clinical trials and diagnostics towards that, which are important and improve the clinical care of LBD. One of the other aspects of the complexity of LBD is that biologically it's heterogeneous, meaning that the biology or what's happening under the microscope in the brains of patients with Lewy body disease is different from different patients. And this is a summary of a multi-center autopsy study we did a few years ago, looking at patients who had early cognitive impairment or DLB and patients who had Parkinson's disease dementia, or who had Parkinson's disease that was established with a varying time interval to develop cognitive impairment and dementia. And this plot is what's called the motor dementia interval, of the time from getting motor Parkinson's to getting cognitive impairment. So as you go up the y-axis here to higher time intervals, you have longer time of Parkinson's with with later occurring dementia, whereas negative numbers and close to zero is the clinical syndrome of DLB. And what you can see here is each dot is an individual brain donation patient who had varying amounts of Alzheimer co-pathology. So the colder colors, blue and green, represent relatively pure alpha-synuclein that's depicted here. So we're not seeing plaques and tangles, and relatively sparse cortical Lewy bodies. Whereas patients who had the redder and the purple had more of mixed pathology, as you could see here with the plaques, tangles, and a higher burden of Lewy bodies. So we see this spectrum between Parkinson's disease and DLB, but we also see a spectrum with Alzheimer's disease. And this is a pie chart from brain donation patients at Penn who had Alzheimer's disease clinically, a memory problem. They didn't have the features of DLB. And what you can see is that more than half of them had alpha-synuclein or Lewy body pathology. And we know that from several different studies. And when Lewy bodies are present in addition to plaques and tangles in patients who clinically have Alzheimer's disease, they tend to have slightly different clinical presentations that can somewhat resemble DLB. So the complexity of the biology of the brain that we can't easily see under the microscope has been a challenge historically, but there's been some major breakthroughs that help us understand in a living patient what the cause of their symptoms is. And what's highlighted here are two main new diagnostic tests that are being developed and are starting to become clinically available, but are largely in the research realm now, to detect evidence of alpha-synuclein Lewy pathology in living patients. On top is one mechanism through a skin biopsy, and you could see here depicted different areas of the skin where a small biopsy of skin can be obtained. And using histology or looking under the microscope, we could see nerve layers. And with a really clever and robust method of using double fluorescence, essentially using two different markers to stain the tissue, you can see co-localization of very small amounts of alpha-synuclein, the same protein that causes Lewy bodies, in peripheral nerve tissue. And we'll talk about some of the studies now that have been going on with this data that help us understand who is likely to have Lewy body in the peripheral tissue is likely to have it in the brain. Uh, Different approach, but similarly can detect Lewy bodies that are in the brain, requires spinal fluid. And spinal fluid is easy to obtain, is a straightforward procedure in most patients. And in patients who have Lewy bodies, particularly a high burden in the brain, that can be incubated, meaning put in a test tube with what we call recombinant alpha-synuclein that is grown in a test tube. And that doesn't have a structure to it. But when the patient sample is put together with the lab-grown alpha-synuclein, it can start to stick together and form Lewy-like aggregations in the test tube. And you can measure it with a piece of equipment that measures fluorescence. And that can give us a readout in a living patient if there's highly likely to be Lewy bodies in the brain. So these two complementary approaches are now emerging as a means to detect Lewy body during life. And two different groups have recently published frameworks for how to use this information to classify alpha-synuclein disorders. There's a lot of nuance to the two different groups' approach, but the idea is that using this biological data helps us get a more precise diagnosis in a living patient. In parallel to this, this table depicts new clinical criteria for Alzheimer's disease. This is a busy slide, but the idea here is that clinical tests that are now available for amyloid, A beta, and tau for the tangle part of Alzheimer's disease are here. And emerging criteria are now also incorporating alpha-synuclein markers that we just discussed. So there are different frameworks emerging to help us detect and classify different biological subtypes of DLB, PDD, and Alzheimer's disease. There's still a lot of work to be done to help us understand. And an important paper that was produced by Jennifer Goldman and colleagues, many who are part of the LBDA Research Centers of Excellence, helped identify how DLB patients in particular have and early stage disease may not be fully captured by any of these frameworks. And as we continue to get more data and understand these tests and understand their relationship to the brain, will help us further refine and improve our diagnostics and ultimately to improve clinical trials. I also want to briefly raise awareness to another framework that's been proposed. This is the Dementia Nomenclature Initiative. Angela Taylor, who's been a leader at LBDA, was instrumental in this. And this was a public-private partnership that included input from the lived experience of patients who have dementia. And the two main take-homes from this initiative were, one, the term dementia itself may have pejorative connotations, and that be something that potentially we would move away from. And the other is to start to go beyond what's called a clinical pathologic diagnosis. So rather than having one term that means what's happening in the brain and what's happening with the patient clinically, start to separate and link these together. So, for example, a patient with DLB may have what's called a dysexecutive disorder, not a memory problem, but more of a problem with frontal lobe functioning and Parkinsonism. So this is the lived experience of the patient. These are things we can help treat symptomatically and with supportive care. And then with biomarkers, and some of them we'll talk about today, we might be able to detect the biology, alpha-synuclein, and in this hypothetical example, amyloid and tau. So this helps us more comprehensively communicate, both as clinicians, researchers, patients, and families. This past spring, the NIH and NINDS held its biannual or every 2 year ADRD Summit, which is a really important meeting that happens. It's part of the National Alzheimer Prevention Act. And as part of this, you can see here Dr. Kathleen Poston, Jim Galvin, and a panel of experts on Lewy body and patient advocacy, including LBDA, were part of the subcommittee for Lewy body specific research priorities. And what is great for multiple reasons about this event, public input was solicited, and it was both in a written format and also during a 3-day virtual meeting where as a group, collaboratively, we designated the highest priorities for Lewy body research. And you can access this online, and I'll have the link at the end. But I want to bring attention to one of the major changes was putting to the top of the list a priority number one is to expand clinical trials for LBD. And I think this is important for so many reasons. But one, for the scientists who do the work, the hard work of mechanistic studies that give us the new treatments that can take years, and this helps put a frame of perspective of helping to accelerate getting things from the bench to the bedside, is what we call it. So hopefully this gives a good overview of some of the terms and definitions and some of the things that we're seeing change in Lewy body, both in clinical care and in research, and the terms that we use. And this will help us as we go through some of the research updates in our year in review. So as we discuss clinical trials and some of the new clinical trial data that we've received in the past year, the first I'd like to highlight is neflamapimod, which is a study drug that has a mechanism of action that is specific to brain cells that produce acetylcholine. So the way that our brains are designed is that we have a small number of cells that produce neurotransmitters or chemicals that then affect the functioning of more broadly in the brain. And the basal forebrain, or an area in the base of the brain, produces this chemical that helps facilitate brain function and cognition. And this paper highlights the preclinical data, the work in the mouse models and so forth to help develop this mechanism. And there was a phase 2 study that was performed that in patients with mild to moderate Lewy body disease. And the main outcomes were looking at different neuropsychological tests of cognition and a global scale called CDR that looks at overall severity of cognitive impairment. The main outcomes that were found in that were some improvements in outcomes of attention and dementia severity and gait. And interestingly, as part of that first study, blood biomarkers of Alzheimer's disease were included. And it was illustrated here after they looked at the data, the clinical benefit. This is showing that a lower number, meaning less of a difference between a higher score for the CDR was seen overall. But when we look at the subgroup of patients who had Alzheimer's biology, here compared to those who didn't, the effect was larger. So, as we discussed earlier, the heterogeneity or the different biology, there's different types of Lewy body disease. Those who have mixed pathology seem to have less of an effect to this drug. And earlier this fall, we heard results from the sponsor of this trial from a follow-up study called Rewind LB that included a larger number of patients with mild DLB, and in this time used the blood test to include only those who didn't have evidence of comorbid or mixed pathology. There was some nuance in terms of the production of the study drug, but overall, those who had the study drug preparation that had the most robust effect of the study drug in the blood showed similar clinical improvements and some element of biomarker engagement with a different blood test. So this is encouraging results that have been provided by the sponsor, and we'll learn more in the future. A different drug called CT1812. This is a drug that is a small molecule, meaning it's a small protein with a specific function to block a receptor in the brain. And it's highlighted in this paper here that receptor interacts with aggregations or oligomers, what we call it, of alpha-synuclein in cell models. So by blocking this receptor, it helps reduce the damage from Lewy bodies. And this trial, the results were released publicly, showing in a phase 2 trial in patients with mild to moderate DLB that had several primary outcomes across the different clinical features of LBD, that in particularly memory and fluctuations in caregiver burden, there were some positive signs compared to those with placebo. So more encouraging news from early stage trials in DLB. It's important to also highlight that these trials were also partially funded by NIH research projects, and LBDA research centers of excellence were also sites where these trials were conducted. So important developments to help accelerate clinical trials in DLB. Another study drug that I'd like to raise attention to is Ambroxol. This is a expectorant that in screening for different compounds that have activities related to Lewy body disease was found to enhance what's called glucose cerebrosidase activity. This is an enzyme or a protein that acts on a specific aspect of the breakdown of Lewy bodies in the brain, and it's the genetic variation in that gene or that protein can help is a risk factor for PD and DLB. So by boosting the activity of that enzyme, that may help alleviate Lewy body pathology in the brain. And one trial in Parkinson's disease with a phase 2 trial that enrolled patients with PDD or Parkinson's disease with dementia, mild to moderate, that included a placebo comparison. They looked at safety and cognitive outcome. What they found was it was safely tolerated, and there was some element of target engagement, meaning looking at blood tests for the ability for to boost that enzyme activity. They saw it, but the clinical benefit wasn't as clear. And this is an early phase study, and there are other studies being planned or in process. One here called the ANID study that's currently being conducted in Norway, looking at this specifically in DLB. So that was an overview of some of the clinical trial updates. Now I'd like to look at or review some of the highlights of observational research or examples of what we're learning from patient data and samples to help us inform new diagnostics and new strategies for treatments. So our clinical assessment of patients is an important aspect of developing new tests for DLB. And in this study from the Mayo Clinic was a retrospectively looking back through records of close to 500 patients who met clinical criteria for DLB. And one of the things that's very difficult to answer for patients on an individual level clinically is what to expect. And a high priority is finding prognostic markers, things that could tell us who is at higher risk for things getting worse in the short term. What's depicted here is a statistical test showing a hazard ratio, and essentially the further to the right these dots are and the associated error, the length of these lines shows you the greater risk of a reduced survival. And on the left are different clinical features of LBD. And essentially, the presence of visual hallucinations and motor Parkinsonism were associated with a higher risk of progression. And the summation of the core clinical features of Lewy body were also associated with a higher risk of progression. So our clinical tools can potentially help us in understanding who's at higher risk. This is a study from the DLB Consortium. This is an NIH-funded multi-center study in the US, led by Jim Leverins, Doug Alasko, and Oscar Lopez. And this very recent study, who came out this month, took a look at a cohort of close to 100 patients with DLB who had spinal fluid testing, where we could test for positivity of the seeding amplification assay that I mentioned before. So biological evidence of alpha-synuclein in the brain. And close to 70% of the patients in the study had evidence of spinal fluid aggregation of alpha-synuclein in the spinal fluid. And that was associated with more motor progression and loss of smell, things we consider related to Lewy body pathology. And comparison, as you could see here on these plots, of longitudinal data for a cognitive test and for motor clinical features, comparing those who had alpha-synuclein in the spinal fluid with or without mixed pathology. We could see this additive effect of those patients who have both evidence biologically of alpha-synuclein and plaques and tangles having a worse prognosis. So good example of our ability to start to detect and understand what biological measures of alpha-synuclein, tau, and amyloid mean for the future. And these could be incorporated in clinical trials, as we're starting to see in some of the more recent trials for DLB. This is a study led by Kajal Kantarsi and her group at Mayo Clinic that nicely looked at blood-based biomarkers. There are several blood tests now that are some clinically available, most in the research realm, looking at markers of amyloid and tau, as well as neurofilament light chain, which reflects neurodegeneration, and GFAP that reflects microscopic inflammation in the brain. And then this nice study that include patients both with DLB as well as early stages of DLB and healthy controls. They found, as depicted here in these heat maps, that these blood-based biomarkers related to amyloid or plaque pathology in the brain as well as tangle pathology. So blood tests may give us complementary information, relative ease of acquisition compared to imaging, and might tell us about risk of progression. And in this study in particular, the inflammation marker was elevated in those patients with early Lewy body disease. There's also a European consortia of DLB as well that in this recent study, they looked at blood-based biomarkers as well in over 500 patients. 340 of them roughly had DLB compared to Alzheimer's and healthy control. So you can see here, these are box plots of the blood-based tests between groups, and you can see that there is a wide variation showing the heterogeneity of DLB and the mixed pathological features that falls in between, largely between healthy controls and patients with Alzheimer's disease. And what was interesting in this study, all the blood-based biomarkers were associated with worsening cognition at baseline. And in the subset with longitudinal data, we could see that inflammation and neurodegeneration correlated with decline in motor impairments over time. So more examples of how blood tests and combinations of blood tests may tell us what to expect. These are two studies looking at patients with idiopathic REM sleep behavior disorder. So this is a recognized prodromal feature. Patients who have this very specific sleep disorder are at risk of developing PD and DLB. And what's depicted here is patients who were observed over time in this first study from Montreal showed that high levels of blood tests for amyloid and tau, p-tau 181 was higher, particularly those who had a cognitive impairment phenotype or DLB. And in this study, a different marker similar called p-tau 217. And what's shown here is a survival curve, meaning time observed of patients with REM sleep behavior disorder developing either PD with MCI or dementia with Lewy body. So similar information that the Alzheimer markers help us in these research studies look promising to predict who's at higher risk of developing a DLB-like phenotype. Another important development on the clinical side of things from the North American Prodromal Synucleinopathy Consortium, or NAPS, was the development of a scale that captures the severity of symptoms. This is really important for clinical trials that may be targeting um, symptoms of REM sleep behavior disorder of a manifestation of very early Lewy pathology. So developing a scale is complicated, needs to be validated with a gold standard, such as a clinician's interview, and also have the properties to have granularity for use in a clinical trial. Another highlight of a different type of imaging study called functional imaging called FDG PET. This was an interesting study through Mayo Clinic looking at patients with DLB or DLB MCI and a subset that over the time period being observed had progressed to DLB. And what's depicted here is a network of brain regions that show hypometabolism. So DLB often may not have clear cortical atrophy, but instead looking at functional outcomes like FDG PET or how the brain metabolizes glucose can give a pattern of patients who may have synuclein in the brain. And in this study, what was really interesting is they simulated statistical power in a clinical trial, and by showing changes in brain metabolism, plus some of the clinical scales that we're familiar with, it increased the power or the ability to detect a treatment change in a clinical trial. So really nice illustration as we can start to put together different scales clinically and biological tests to make clinical trials more likely to be successful. On the flip side, we talked about how clinical Alzheimer's disease can have Lewy pathology and how that may manifest. This is a study from Penn using the ADNI cohort, which is a consortia study of patients with clinical Alzheimer's disease. And this is a busy slide. What I'd like to highlight is that those patients who have, if we look at their PET tau and amyloid and imaging metrics and find patients who have a disproportionate amount of functional hypometabolism, as you can see here, the cold colors in the back part of the brain that look very similar to the previous study that I showed you in patients who had clinical DLB. This corresponded very well to the spinal fluid test for seeding amplification of alpha-synuclein. So now that we have some of these other biological tests for synuclein, we can start to look at what's the signature in imaging and other features that could tell us if Lewy bodies are present in the brain. So now I'd like to move on and finish with some highlights of mechanistic research. These are research looking at human tissues or model systems to help understand the biology of Lewy body disorders. And one large-scale study that came from a collaboration of Banner Health System with a group in Sweden using a really impressive cohort of individuals who participated in brain and body donation where alpha-synuclein could be measured under the microscope and using both in the central nervous system, the olfactory bulb, which is part of the connection to the brain that helps with processing smell, which is linked to anosmia, as well as peripheral nervous system in the gut. And using advanced statistical methods, they were able to use a data-driven approach, saying the observations from the tissue, what are the subtypes of patterns? Because there's been many different patterns that have been described looking at patients qualitatively about where synuclein is in the brain and then what we call the neuroaxis between the brain and the peripheral nervous system. And you can see these different schematics here that have been described in different studies. And in this study here, you could see these different data-driven stages, some including the olfactory bulb and what's called the limbic system, so kind of bypassing the motor system. And you see it's enriched in patients who have clinical Alzheimer's disease compared to those who have a brainstem and a motor early tend to have more Parkinson's disease, and then there's some subtypes in the middle. So really interesting data showing a spectrum of distribution of Lewy bodies throughout the nervous system that may help us understand why some patients have some symptoms of DLB or AD compared to others. And then another group using similar methods in patients who did not have overt DLB or PD, but had evidence of alpha-synuclein microscopically in the sympathetic nervous system or the parasympathetic nervous system and in the brain. And by looking at all that data with statistical modeling, as you could see here, is just showing you how they could take that data and put things into a hypothetical order. They found a really nice distinction between two subtypes of patients that might preferentially include one or the other of these two areas of the peripheral nervous system. So again, showing us more heterogeneity. And the more that we understand these different subtypes of the distribution of synuclein, that might help us understand mechanisms, genetic triggers, and environmental triggers as well. This was a study recently published in the journal Science by a group at Johns Hopkins, which was a really nice study incorporating both epidemiology data and several different eloquent experiments with mouse and cell cultures. And as summarized in the schematic here, there's a lot of data to unpack, but the overview is that they first started with epidemiological data from healthcare records of over 50 million individuals in the US. And by looking at zip codes of individuals and diagnostic codes in the medical record, they found an association of patients who had particularly DLB or PDD with areas of high exposure to air pollution, something called PM 2.5 as a specific type of particulate matter. So then to go into mechanism, they did a series of experiments. One was by exposing what's called a wild type mouse. That means a mouse with no genetic manipulation. And by exposing these mice to this particulate matter, they were able to detect atrophy and disease in the brain, including alpha-synuclein Lewy pathology. When they genetically then manipulated a mouse to remove alpha-synuclein from the brain, they found those mice didn't have atrophy. So that suggests that alpha-synuclein, the component of Lewy bodies, may be playing a role in how the exposure could potentially affect brain health. And then in other series of experiments, which are very interesting, they expose different formulations of what's called preformed fibrils. These are a way to take patient-derived or other preparations of the alpha-synuclein protein that have already stuck together into what's called a fibril and introduce these into mice. And those, the exposure of the fibrils to particulate matter could help develop a mouse phenotype, meaning the mouse looked more like had more cognitive impairment and other features of DLB compared to PDD. So provocative, interesting studies integrating epidemiology and mechanistic work, suggesting at least a possible contribution of air pollution exposure as one mechanism of developing Lewy body pathology and clinical symptoms of memory impairment. Another example of epidemiology and mechanistic work to influencing alpha-synuclein is two papers I highlight here. One was an epidemiology study in Sweden looking at medical records of individuals who had renal failure of different acuity and severity, finding an association of different types of dementia. And particularly in this study, PDD and DLB had particularly high hazard ratios or relative risk in the setting of renal impairment. And in this series of experiments in this study highlighted here, using mouse models and looking at human kidney tissue, alpha-synuclein was detected in nervous tissue surrounding the kidney, alpha-synuclein in the blood, and the mouse model was able to be cleared from a renal mechanism. And they were able to demonstrate in a mouse that alpha-synuclein could be transmitted from the kidney to the brain, at least showing proof of concept that that could be another mechanism of general age-associated health influencing the risk of developing or accelerating Lewy body pathology. These studies will need continued ongoing validation and further replication, but very interesting and highlights. This is a study from Penn that looked more specifically at blood measures of alpha-synuclein. These are also busy slides. What's depicted, the more that we are learning about alpha-synuclein and how it aggregates in brain cells, there is data to suggest that there may be strain-like properties, or at least different aggregations that have different folds or conformational changes or biological properties that might help explain the differences in phenotypic expression or the clinical features that patients experience. And what's depicted here was by looking at serial passages or putting fibrils, the connections of Lewy body, like pathology into mice, you can generate strain-like properties. 7015 or strain A and 9027 or strain B. In a test tube and in animal models, we see strain A is more associated with Lewy body pathology alone in these cells, whereas strain B can also interact with tau and cause tau, the protein in tangles in Alzheimer's disease, to accumulate. This is brain tissue examples that we stained in my lab and quantified the strain A and strain B and correlated with AD pathology. And we found that the strain B correlated with tau pathology more strongly than strain A, which is interesting and suggests that, these may affect, be implicated in different co-pathology. And then by looking at both at patients from UPenn and a PDBP and NIH database, interestingly found differences between PD and without cognitive impairment and DLB, suggested that differences in strain-like properties might explain why some patients may have a cognitive impairment and some may be less likely to. And what was interesting from this study as well is that by taking patient-derived blood, synuclein was able to induce alpha-synuclein and Lewy-like pathology in a neuronal culture. So brain cells in mouse brain cells in a culture system that keeps them alive, where they were recapitulating Lewy pathology. So this and other data that I've been showing you suggests that there may be peripheral alpha-synuclein that plays a role in LBD as well. And to circle back from the bench to the bedside, this is work from Alice Chempropkin and colleagues with David Walt at MGH, looking at new ways to measure blood-specific tests for alpha-synuclein, something called extracellular vesicles, complicated methods, but to briefly summarize an overview here, in the blood, there are very microscopic and small frequencies of what are called vesicles that can be derived from the brain. And now with new technology, we're able to measure these very sensitive small amounts. And in this study, provide proof of concept that there may be elevations or at least relative higher alpha-synuclein in these brain-derived cargo packages compared to the rest of the blood. And in a different study focused on Parkinson's disease, using slightly different methods to detect extracellular vesicles, they looked at patients with REM sleep behavior disorder who are at risk of developing PD and DLB, and found, compared to healthy controls, that there was higher alpha-synuclein in these vesicles. So helpful illustrations of what we learn in the brain can eventually potentially translate into diagnostic tests that we can get in clinic. So I'd like to end there to summarize that we're seeing an acceleration of LBD research, and the focus is now really clearly on diagnosis and treatment. We're learning a lot. It can't happen fast enough. And part of that is a growing national and international collaboration. Team science is really important, and multidisciplinary science. Some of the work that I showed you involved people with very different disciplines all working together to answer a fundamental question. And we all learn so much more from that approach. And particularly for LBD, where it can manifest with so many different clinical features, having people from sleep background, psychiatry, movement disorders, cognitive neurology, and mechanistic scientists who can work with animal models and cell models and epidemiology is really, really important. And then, a special thank you to those who are able to participate in research, including brain and tissue donation. It's a deeply meaningful contribution, and hopefully I helped highlight the impact from all the folks who participate in studies like this. What do we expect for 2026 and beyond? I think we'll see more clinical trials with patient selection criteria that are more addressing the heterogeneity and the different biological subtypes of LBD. We'll see more data on diagnostic tests, including blood tests and things that may be quantitative for alpha-synuclein. And looking at more expanded mechanisms such as inflammation in the brain, more on exposures, the lived experience, and genetics. So I'd like to end there now with highlighting some resources. LBDA, of course, has excellent resources, and you'll hear from Keith at the end, as well as news updates from our center at Penn focused on alpha-synuclein and Lewy body. PubMed is a public way to query for medical literature. Al's Forum, which has wonderful summaries of clinical trials and recent publications and scientific meetings, which is very accessible to both scientists and patients and families. And then clinicaltrials.gov to find clinical trials and also very helpful resources on the what learning more about clinical trials from the patient perspective. So I'll end there and look forward to discussion.
All right, thank you very much, Dr. Irwin, for that engaging presentation. We are going to take about 5 minutes here for some Q&A. Oh, that's great. Thanks to everybody for using your reaction buttons. Appreciate that. We won't have time for a ton of questions, but I do want to put a few through, especially for there were some themes that came through during the talk. So I'm going to start with something actually a fairly straightforward question, which is about hallucinations. And the questioner was asking, could you describe, what counts as a hallucination in LBD? Is it, seeing something that isn't there? Are we talking about flashing lights, rainbow colors, et cetera? What would be a true hallucination?
That's a great question. And one of the more challenging core clinical criteria for DLB for us to detect clinically, and it often requires a good historian, the patient, and sometimes family members. Um, uh, the type of hallucinations we think of from DLB and not from a medical source would be spontaneous, meaning it's not associated with a delirium or a general medical condition or drug. So that would be first to exclude those precipitating factors. They're usually well formed. So people, animals, things that are can be quite vivid and detailed, like a specific person or animal. And patients may describe a similar recurring hallucination that they see and persistent. They have to not be once or twice, but something that's persistent. So things are less well formed, like flashing lights, and things may be a sign of another type of either brain pathology or process neurologic condition or something non-specific. And it sometimes early can be manifest as something called an illusion. That's when we have a visual stimulus like a carpet pattern or a wallpaper that can induce a patient to perceive something that's not there. I see a face in the snow, or I see a face or an animal in the pattern in the carpet. That's not a core criteria, but that's incorporated in different neuropsychological tests for DLB. And then also important to help tease apart from what's called delusions. So these are false fixed beliefs. So sometimes patients of all types of neurodegenerative dementias could have a belief that maybe someone's in the house stealing from them when they're having trouble with memory. And that can be hard to tell apart from a true visual stimulus. So the classic story is someone really describing vividly someone who is not there or an animal that's not there. Usually it's not distressing, but it can be. And that's something, of course, to go to your doctor, physician, and medical team about for help and support.
Great, thank you. I'm going to actually leave us with the sort of best question, if you will, that came through. This is one that has chatting backstage going, oh, we have to ask this one. So someone asked if you could wave a magic wand and fund any study that does not currently exist, what would it be? And feel free to mention something that you've submitted as a grant proposal, maybe.
Yeah, no, that's a great question. I think, the low-lying fruit would meaning something that we could learn a lot from that would be low risk. I think in terms of impact would be there are amyloid therapies that exist today that are clinically indicated for Alzheimer's disease. We don't know what the safety or the potential clinical benefit would be for patients with DLB and mixed pathology. So that would be something that data I would love to see. Um,
Great answer. Yeah, I think the whole community would like to see that. And for the those who are listening, this harkens back to something that Dr. Irwin said earlier about mixed pathology and how many people who have Alzheimer's also have Lewy pathology in their brain, and vice versa. How many people with Lewy body disease also have amyloid pathology in their brain? And so I think the whole community would love to see, is it possible to treat people with LBD with one of these new medications against beta amyloid, which is more commonly thought of in Alzheimer's. We're going to let you go. Dr. Irwin, thank you very much. Appreciate your time here today. And for the other folks who ask questions, we'll try to answer some of those by writing as well after the webinar today. So unfortunately, we are out of time. I do want to mention a couple of things before we let everybody go, though. You heard a lot about clinical trials today, and I want to let you know that there are at least two ways that you can learn about clinical trials through LBDA. The first and easiest is to sign up for something called a Lewy Trial Tracker. Lewy Trial Tracker. That is essentially a contact registry for people who want to learn more when new clinical trials launch. Essentially, you go to the website, lbda.org, you navigate to find the Lewy Trial Tracker page. You can just search on our website, and then you complete a brief survey. It tells us a little bit about you and your research interests. We then use that information that you give us to tell you by email when a new study launches that fits what you told us about yourself in that survey. So it's a really nice, convenient way for you to ensure that you get notified whenever a new study starts enrolling that matches your interests. A couple of important notes about Lewy Trial Tracker. I think most importantly is it's not a commitment to join any research studies. It's really just a way for you to learn about new studies as they launch. And if something turns out to be of interest to you, then you can reach out to that study team. But the study team will not reach out directly to you because critically, and this is the second thing I want to say about Lewy Trial Tracker, is that we never share your information with study teams. We only use your information to figure out which studies to make you aware of. And then it's up to you, if they are of interest to you, to reach out to that study team using the information that we will give you about them. But we won't share your information with them. The other way that you can learn about new research volunteer opportunities is through LBDA, is to occasionally check the clinical studies page on our website. Our aim is to list every study that is LBD related, currently seeking volunteers, and open in at least 2 locations. We also list numerous online studies that can be completed from anywhere. So you can complete these studies from the comfort of your own home. Each entry in those study listings contains locations and contact information for the local study team so that you can quickly see if there is a site near you, and if so, who to contact for more information. With that, I hope you all found today's webinar informative and enjoyable, but unfortunately we are out of time. On behalf of LBDA, I would like to thank you all for taking the time out of your day to join us, and thank Dr. Irwin again for sharing his expertise with us. As a final reminder, you will receive an email in about 1 week with the link for the recording of today's presentation, as well as the resources and links that were mentioned during the webinar. And please do take a moment to complete the survey that will launch when you leave the webinar today, because as I said before, that truly does help us inform our content and keep it relevant for you. With that, have a safe holiday season, and we hope to see you again in the new year.